So what we're going to do now is we're going to do the vision lab. A lot of this is going to be stuff you're going to have to do at home. We actually do have a lot of little things that will actually allow you to do what you want to do what we're hoping that you get to see, but you're not going to do it in lab with your lab mates. Instead, you'll like print out a little, Snellen eye chart and stick it on the wall at home and shut an eye. And ultimately, it's going to be better if you have somebody doing it with you. Some of these, again, are going to be tricky to do totally on your own. Some of them will be easy to do on your own. Some of them will be about watching videos but still writing in your lab book what your experiences are in the video. Some of it will be more kind of hands-on, so what I'm going to do is I'm going to give you background. I'm going to talk about the basic background on the eye that you're going to need to kind of make sense of this lab, and then kind of go through the different parts of the lab so you have a good sense of kind of what the expectations are and how we're going to kind of adapt them to kind of the stay-at-home kind of thing. So obviously it's going to be about vision. What's the stimulus for vision?
Light.
It's light, electromagnetic radiation, which comes in different frequencies, just like sound comes in low frequencies and high frequencies, right? There's a whole range from in terms of sound. For light, the vibrational frequency of the electromagnetic waves we perceive as different colors. The lower frequencies are red. The highest frequencies that we can see are up in the violet, right? So, but it's just how fast things are vibrating, except our subjective experience is color rather than pitches of sound. What's the main organ that actually accomplishes the sensory transduction? Well, this is an easy one.
Your eyes?
Your eyes, right? Your eyes are going to be the things that convert light energy into some electrical signal for your brain. Your eyes are doing two big things, though, and we're going to be looking at both of them in this lab. So if this is your eye, this is like a cross-section of your eye. Like if I was looking at the eye from the front, you would see there's the pupil, there's the iris. But if I do cut a cross-section through it, what I would see is there's like the cornea, which is kind of the clear window going into there. The iris kind of comes down like this. Right? If you think about a Frisbee with a hole cut in it, if you do a section right through it, this is the side view of a Frisbee with a hole cut through it if we go right down the middle. And right behind that, we have the lens. And the lens is actually, there's the ciliary muscles and things that can adjust. We'll talk about, you can actually adjust the lens. So the iris and the lens are about kind of getting a good image. It's like on a camera, you have an iris and a lens. You can adjust the amount of light coming in by making the iris larger or smaller. the lens, you change the curvature of the lens to bring something into focus properly. But none of that is actually doing sensory transduction. The iris and the lens are about getting a good image in focus. But then we also need the part of the eye that actually converts the light into an electrical signal, and that's going to be the rods and cones of the retina. Low battery alert, low battery alert. So the retina with the rods and cones, that is the place where the actual sensory transduction happens. I think I think in the lecture we'll talk much more about the details between rods and cones and all that. Right now, let's just say rods are only detecting kind of absence or presence of light. They're called what we call monochrome. They are much more sensitive to dim light. The cones are the ones that are for like color and high acuity. In fact, let me, and most of the cones are located right in the back. So this is called the whole area is called the macula lutea, but the main thing is called the fovea centralis. The fovea centralis, when you stare right at something, the image lands on the fovea centralis. This is where you have these cones which are very high acuity, meaning they can tell really good detail. This is all cones, high acuity, and color vision. Most of the periphery, kind of more along the side, tends to be the rods. It's actually a really kind of weird thing especially when we think about cameras. You think about like your iPhone has a 12 megapixel camera now, and that's probably going to be obsolete. Your eyes, at best, if you think about the density of rods and cones and stuff, it'd be like a 1 megapixel camera. It's really stinky in terms of, a camera like on your phone or something. But it's got really good detail right in the center. But as soon as you get off the center, you're in this low acuity rods. It's like if you look straight ahead, you can see a lot of detail. If you put your hands to the side of your head where the image is landing more on the sides of your retina, you don't even know how many fingers you're holding up, right? The only thing you actually see really in high acuity is what you're staring right at. We actually even have a verb. We say foveate when you stare right at something because it's landing on the fovea centralis. Even more kind of trippy is the way the rods and cones in the retina are hooked up. All of their axons are coming up, and then they have to kind of burrow out the back to go to the brain and the optic nerve. So you have this place where all of the axons have to burrow out, and that's going to become this area called the optic disc or the blind spot. So if you're staring at something and that image actually lands on that part of the retina, there are not even any rods and cones to do sensory transduction. So you have no idea at all that there's anything there. Obviously, when you look around, you don't actually see this big empty space everywhere you look. And so your brain will make stuff up. We'll talk about this blind spot in just a few moments. In terms of rods and cones, let me talk a little bit more, particularly about the cones. Like color, you're looking at the screen right now and you see the blue, you see the red. The reason why you see those colors is because you have particular cones that respond to particular colors. So if I look at I'm just going to have this general response. And here's kind of frequency of light from lower to higher. You're going to have red cones that respond to these lower frequencies, these green cones that respond to these mid-range frequencies, and blue cones that respond to the higher frequencies. And depending which population of cones are being activated in what proportion, that's how you see different colors. If you look, when you look at a plant and it looks green, why does it look green?
Isn't the green like bouncing off?
Exactly. Like the chlorophyll is absorbing all the red to do photosynthesis as well as the higher frequencies. So the kind of the waste light, the light it's not using, is the green. So that's bouncing off and entering your eye and you see the plant is green. Right? If you see something that's black, somebody's super cool walking around their black turtleneck, why does their turtleneck look black?
Is it because the black is absorbing all the colors?
Yeah, exactly. There's no light coming back, so you're not stimulating any of the receptors, so it's black. And then white, and a little counterintuitively, if you've just been thinking about pigments, white is just the mix of all the colors. Right? When you're in elementary school and then you're mixing pigments like paints, you have like green comes from yellow and blue, and purple comes from blue and red. But if you mix all the colors together in your paint bucket, it's just going to look like mud. I'm sure you've done that, thinking it's going to be really cool when you were a little kid, and it's like, oh, that's so disappointing. But with light, mixing luminance is very different. If you mix all the colors of the rainbow together using luminance, using kind of glowing light, you actually get white, right? That's why if you take sunlight and you break it apart through a prism, you get the whole rainbow, because all those colors are in white light, just all mixed together, and the prism separates them so you can see them separately. So it's a little non-intuitive if you just think about pigments, but using the red, green, and blue, you can mix to make every color. Some people are born with a genetic problem where one or another of these cones don't get made properly. Like, so let's say somebody didn't have red cones. What would that mean?
That they can only see green and blue light?
Exactly. They're only going to respond to these. So anything that is differing by some amount of red light is going to look the same, right? If I have like something that to someone with normal color vision is like, what are you talking about? That's like reddish green. That's like kind of reddish brown, and that's more of a gray brown. And person who is lacking these red cones is going to say, no, they look exactly the same. I don't see the difference, because they don't have the ability to detect the addition or not of adding the red into the mix. So that's color blindness. Color blindness is when you do not have one or more kind of cones. It's sex linked. What does it mean if you have some sex linked trait, color blindness?
Isn't it for your mom?
And how is
To
that?
the man or to the boy?
It's not actually, not necessarily through your mom.
No.
It could be from your dad. But it's most.
But doesn't it come out in the male?
It means it's on the X link. It's on the X chromosome. So the code to make the opsin for these cones is on the X chromosome. You know, what that means is guys are way more likely to be colorblind, right? Right? Because if you're a woman, you're XX, right? Everybody remembers that from biology. You're XX. So let's say you have one bum copy of the gene to make the cone. You've got a whole other X chromosome from your other parent where you probably have a good copy, so it's all good. If you are a dude, though, and you have a bum copy of that gene, your other sex chromosome is a Y. So you do not have a backup copy. So it's way more common to have color blindness in guys than women, because women have two X's, so they have two chances to get a good copy. Whereas in a guy, if you have one bad copy, that's it, because your other sex chromosome is not an X. In teaching this lab for like 25 years, fairly regularly we see students, males, actually usually way more women than guys in the class, and yet we still see a fair amount of color blindness amongst the guys that come in the class. I've never ever seen it exists. You can have a woman with two bum copies of the X, but I've never seen it. If you want to test if someone's colorblind, there are these special plates, the Ishihara color plates. So actually at this point, it probably makes sense. I'll shift gears a little bit. I'll show you the parts of the lab where we're going to be looking both at blind spots and looking at the color, kind of the stuff that's more about kind of the retina. And then we'll shift gears and we'll talk more about the lens and stuff, about the focusing mechanics of the eye. So let's see, move here. All right, so this is the lab here. Special senses vision. Demonstrating the blind spot. The main way you demonstrate the blind spot, I have here, you can do it in your lab book. So, for your write-up, you can either just follow the thing in your lab book. Does everybody have the lab book in front of them right now? So you should do this, especially if you've never done it. It's really trippy. Basically, you're gonna, it says hold the page about 14 inches from your eye. You basically are going to close your left eye, look at the R with your right eye, and then move the page closer and closer, but notice the L. Stare straight at the R, but notice the L. And what you're basically doing is playing with the angles until that L lands on that part of your retina that is the blind spot, where there's no rods and cones. So I'd like to see everybody doing this. And there's it's a subtle place. You want to kind of bring it closer and closer. If you go too close, it's going to be on the other side. It'll be back in where there are receptors. But so raise your hand once you get to a place where the L disappears. You can raise your hand with the little hand raiser deal under reactions. Right, I just did a reaction. Oh, so Grace has got it. So Lindsay, probably slow down. You're going fast enough that you're going to miss it if you get it. So again, stare at the R, but notice the L. At some point, you should just see it completely just disappear. It's not subtle. It's like, holy crap. That's weird. And you need to have one eye closed. It's not going to work if both eyes are open, right? And again, move. So I see a lot of people have seen it. So who hasn't? I see Marbeam. Have you not seen it yet? Leah, Alexia?
I still see it.
So are you shutting your left eye? Just use your right eye and look at the R. Wait, so that's your left. No, so right. Yeah, exactly. Now look at the R and bring it closer and closer, but notice the L.
Oh yeah, it disappeared. Yeah.
Here's right. So I want to keep moving. If you haven't seen it before you're done, I want to make sure you don't just move on and write up your lab without actually experiencing it. In fact, we're going to talk. So what is happening there? Why is it happening? Why does that happen?
Well, it's the blind spot, right? It's hitting a spot where it hits your blind spot.
It hits your blind spot, and so your brain has no information about what's there ever. So your brain is just always kind of wallpapering over it, using whatever kind of makes the most sense, kind of pattern completion, right? If it's on that page, it's white everywhere else. So what are the chances it's not just white right there? So it just completes the pattern by making it white. But you can actually have more complicated pattern completions. Like, you don't have to do it now, but later on I have where you can look at a wheel, and if the spokes converge on the blind spot, your brain will just say, of course it's just gonna, I'll see the spokes converge. Or there's a line where it's actually missing a chunk out of the middle, but if that chunk lands on the blind spot, your brain will just complete the line, because that's what makes the most sense. In this demonstrating the blind spot under doing the lab, I have some videos which are kind of fun. Here it says YouTube video on blind spots with groovy music you can do. Oh, wait, where's the groovy music? Oh, come on. Oh, dag nabbit. There it is. Right, so not only does it, so this, it starts with this, but then it gets deeper. It says, try it again with colors. You know, you're gonna find you'll just wallpaper over with the right color. You know, it gets even further. There's a line. If this weird dot ends up on your blind spot, you don't know what's there. You're just gonna have a line continuing on. You know, so play around with this. Here, if this thing lands on your blind spot, your brain's just gonna complete the pattern. What's the chance it's not just another red dot? You know, so play around with this, and you know, in your lab notebook, write down what you experience and why it happens. So, you know, and in reality, both eyes see slightly different views of the world. So you will actually not, you know, be as lost as this. You'll have two eyes to actually double check that you're not totally missing something. But, you know, this part here, this little thing is pretty cool. This one here from Subaru is more talking about the theory behind blind spots, but it's pretty good as well.
Something better for myself.
I was starting to get comments from well-meaning people that would say to me, I don't understand how you could.
Yeah. If you haven't been watching YouTube, you probably think that exercise is actually good for you and will help you stay in shape, but actually it's all a lie. It's like you need to just follow their thing about a special thing to eat, and that's what you really need to do. It's weird, I have to say. So this guy here, he talks about blind spots. He's got like this, he's got a weird little like snarl, but he's actually he talks about this stuff well. I think he's intense. So anyway, you can go back.
This episode was proudly made possible by the all-new 2015
Subaru. So Subaru they've done some good here. After images. So I'll get to that in a second. Testing for color blindness. This is something you're going to need to do at home. The way this works is normally if we were in lab.
It's not that easy. But Grammarly can help. This sentence is grammatically correct.
They also, I don't know if you get them, Grammarly. They never stop. They're trying to like make you like feel like writing is so hard. You better use our app. It's like, don't trust them. You know, you can get good at writing yourself. You can like learn like vocabulary. All right, this guy.
Are you colorblind? Well, in this video.
Yeah, this is kind of. He's also he's an intense looking guy. He has this Ishihara color plate book, which is what you would use if you were going to test someone for color blindness, and it has all these different plates. And these dots are designed so if you have color blindness, they look like one thing. If you don't have color blindness, they look like something else. Like this one here on the left here looks like a 7, and the one on the right looks like a 45 if you have normal vision. But if you're colorblind, you would look at this and you wouldn't see anything. Like if your red cones didn't work, you couldn't see the difference between these dots that make the 45 and all the rest of the dots. So it would just look like a bunch of pebbles, right? So by looking at this and telling the tester what you see, you can tell if somebody's colorblind or not. Normally, again, in lab, we actually have these books. They're expensive. They're like a few $100 to get one of these little testing books. But this guy made a nice video where he just goes through. It takes a little while, but he just flips. Look, he's so intense. And his eyes match his shirt and his tie. So I think, but he goes through page by page. And describes what you would see in one case or another case. So what I want you to do as you watch this video is what do you know, this one here, this is like a control one. Anybody should be able to see this. You know, this one, though, this will look like a 3 if you have color blindness, will look like an 8 if you don't, right? It really depends. So you're going to go through this, and in your lab notebook, you are going to report for each of the pages what you see and what it means. And again, I'm not going to go through every detail with you because we don't have the time, but basically I say, look at the following Ishihara color plates, record your results and interpretation in your lab notebook. There are a total of 14 plates. So for each of those plates, as you watch that video, and if you are a woman, it's probably going to be pretty straightforward. If you're a guy, there's more of a chance that you're going to actually get like kind of a disconnect, but between what most people see and what you see.
If we're a woman, can we do it on like a man and see how they respond?
Yeah, that would be great.
Okay.
Yeah, that would be more interesting. Yeah, usually when we do this in lab, I say if there's a guy in your lab group, it's more interesting. You're more likely to see something interesting if, yeah, no, this is when my uncle's colorblind. I remember asking when I was a little kid, like, how do you know if you should stop or go at the stoplights? And he's like, duh. It's like, I'd never thought about like there's just an order, like the greens on the bottom, the reds on the top, you know. But when you're colorblind, you think about stuff like that. All right, after images, so maybe I should stop sharing for a second. So after images happen because we have, let's say, some rod or a cone. So we have light coming in. And it's sending messages out to the next level. You can kind of overdo it. If you just kind of keep going, let's say this is a red cone that actually detects red. And you stimulate it really strongly with red light. And after a while, it kind of poops out. It's going like zit, 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 zit. And then it's like, I need to recover. So what happens is when you take away this stimulus here, not only is that original red not there anymore, but this thing here is kind of recovering, and you kind of have a lack of red in that area. In that case, in complementary colors, this is going to actually, this part of space will look kind of like greenish, because not only, so you're responding to red less than normal, and if things have less red, they actually start turning green. And again, you have to know just like how light luminance works. But basically, red and green are complementary to each other. Like if you take red and green are kind of the two sides of a coin in luminance. So if I have this red cone that I have been overexciting and now it's kind of resting, that part of my visual field is actually going to look kind of greenish until that red recovers and balances the red out again. If that makes sense. So that's kind of this after image, or what we call a negative after image. Actually, there's two kinds of, I'm being a little confusing here. So there's two things that can happen. One is if you have a really bright light. Let's say you're looking at this, and this actually happens, you're looking at the sun setting. I'm sure you've all done this. You're like, oh my God, it's beautiful. And I'm staring at the sun, even though they always told me don't stare at the sun. And then all of a sudden you're blinking and everywhere you look is a freaking sun. that's called a positive after image. This thing has been stimulated so much, it's continuing to send out messages even when there's no stimulus anymore. But then it gets to this negative after image, and now all of a sudden where you saw this red sun, you're actually seeing kind of like a green dot. That green dot is the negative after image of where the big red sunset was, because now those cones are having to recover, and while they're recovering, everything looks kind of green where they used to be responding. So this is pretty common, where you have a bright thing. First, you see a positive after image where you notice something there even when it's not there anymore. And then you start seeing the opposite color of what used to be there. Or if it was white, you see actually kind of a darkness. If it was green, you see a redness. If it was red, you see a greenness. So that just has to do with kind of overexciting the receptor and then kind of leaving a lack of response for that particular color for a little while. So there is a really cool thing that you can see this negative after image. I think we can probably do it on your screen. I'm going to share the screen again. So this is after image. This is another YouTube thing that's pretty awesome. And you can work along with me. So after image demo. So basically does everybody see this? There's a little purple dot. It's important when you do this that you really let the image rest on the same part of your retina. So you're going to see a picture kind of with opposite colors of a bee. You just keep staring right at that little purple dot while the picture of the bee is on, and it'll last for 30 seconds. And when that's done, the bee is, the negative vision of the bee will disappear. So keep staring at this. And again, this is like all the opposite colors that a bee should be. But what it's basically doing is kind of getting all of those cones kind of tired out. We got another like 10 seconds. And keep your image right there and notice what colors you see. It's going to then just switch to actually a black and white picture of a bee, but see what you really see. Did everybody see anything weird there? What color did it look like?
Almost like yellow.
It looked like a color bee. Like if I back up slowly, black and white, black and white, black and white, right? The difference between when the thing turned off and when you went to the picture, it was just black and white bee. All the color that you saw for a few seconds there was that negative after image of, where the reds were tired out, things looked more green, where the greens were tired out, things looked more red, etc. So again, in your lab notebook, kind of do this and describe what you see. So for part 1, part 2, and part 3, just you are still responsible for kind of recording in your lab book kind of observations, and describing what it means in terms of, photoreceptors and the retina and stuff like that. So that all makes sense? So the other parts of the lab are going to be more about the focusing part of the eye. The lens, and how do we bring something into focus? What is visual acuity? What's astigmatism? What's accommodation? We'll talk a bit about binocular vision a little as well. But maybe we'll do is take a little break. Maybe it's 11:15. At 11:30, we'll come back, and then we'll focus in on the other part of vision, which is the, again, the visual acuity and the lens and all of that. So let's take a little break, and we will continue with the eye.
I have a quick question. So we only have one blind spot per eye, correct? Where all of the
Yes.
things are exiting? Okay.
Mhm. All right. All right, give me a couple of moments here. Okay. All right, so what we're going to do now is take a look at the focusing part of the eye. So let's do that. We need to do a little bit of background with optics and talk about lenses. So let's do that first. If I have a lens that kind of bulges out on either side, we call that a convex lens. And a convex lens bends light inward. Like if I have my light rays coming in, they are going to get bent, so that we call this converging. Because the light rays get bent inward. The more kind of fat the convex lens is, the more the light will bend. If I have like even more of a bulge, then the light rays will come in and they'll bend even more. So, and in our eyes, our lens actually has the ability to change the amount of bending that the lens does. We have little muscles, the little ciliary muscles holding on to the lens with the little suspensory ligaments, and by relaxing or pulling more, you can change the curvature of the lens, and therefore the light rays will bend more or less. That's going to be at the core of bringing the image into focus, is by changing the curvature of the lens using the little ciliary muscles in the eye. The other kind of lens that you need to know that's not in your eye, but is used in corrective lenses, so you should understand how it works, is a concave lens. Concave is where they kind of suck in like that, and this will make the light rays diverge, meaning move outward. When I was trying to remember concave versus convex, I was like, again, if you were like a bear looking for a place to hibernate, you know, you'd kind of have to go, you'd go in here, right? This is like a little cave. You couldn't, it's like, there's no place to hibernate in the convex lens. So that's helpful. I don't know. But we're going to see this, we're going to start with this convex lens, and let's put it in the context of the actual eyeball now. So there's the retina on the back, which again, you can kind of think of as the film in a camera, right? And the film is going to get exposed if there's light, or won't get exposed if there's no light. But unless the image is in focus, you can take a picture and it can be kind of useless because it's all blurry and out of focus. So we need to make sure that the image landing on the retina is in focus. And that's going to be the job of the lens. So here we have the lens, and again with the little suspensory ligaments and ciliary muscles that can change the bending to make it bend, converge more or less. Here's my lens. I'm going to first describe what's called the Emmytropic eye. The Emmytropic eye, this means kind of a normal vision eye. This is an eye that doesn't need any glasses or anything to have 20/20 vision. So this is kind of an eye if everything is functioning properly. If an eye is functioning properly, any light rays coming in parallel, and this is anything from 20 feet and further away, you can kind of assume that the light rays are coming in parallel. The bending of the lens when the eye, and this is like the eye is relaxed here, will be just the perfect amount. And I'm being a little fast and loose with my ray diagrams. In the reality, the image is turned upside down and there's more complicated stuff going on with the optics. But let's just say that the lens has just the right amount of curvature to bend the light rays so they land right on the retina. And let's say I'm looking at like some tree off in the distance here. That tree is going to be landing on the retina just in focus, and I will see the tree in focus all nice. If I'm trying to look at something closer, let's say there's some flower which is right next to my eyeball. Let's say this is just a foot away. If you have something that's really close, then you no longer have this approximation of parallel light rays coming in. The light rays are basically diverging by the time they hit the eye, which means if the lens just stayed the same, it didn't bend anything different, the amount of bending that the lens would do would not be enough to bring these into focus on the retina. So instead, I have to do what's called accommodation. Let me draw that better. Accommodation, my lens is going to get more rounded. That happens, it's actually an active thing. The little muscles will contract, release tension on this lens, and the lens bulges out a little more. It's a little non-intuitive. When the muscles are relaxed, there's tension that kind of pulls the lens flat. When the muscles tense more and accommodate, then the lens can bulge out. When the lens bulges out more, that bends the light rays more, and then they land here, and now my flower is in focus. Right, so that's when something is far away, the eye is relaxed, and the lens has just the perfect amount of curvature to make it into focus. When something is close, you have to accommodate, which means you need to make the lens curve more and bend the light more, but ultimately bring it into focus. At some point, if something is really, really close to the eye, I don't know, let's say, I don't know, I'm just putting a little black star here. If it's really close, the light rays are now really diverging by the time they hit the lens. And you can get to a point where no matter how much you try to accommodate, there's a limit to how much extra curvature you can have with the lens. So at some point, something is so close, you no longer have the ability to accommodate, and it's just going to be blurry no matter how hard you try to bring it into focus. That's called the near point of accommodation. This is basically the closest you can bring something and still pull it into focus using your eyes. If you're any closer than that, no matter how hard you try, it's going to stay blurry. Right? You've probably experienced that. You bring something close enough, and though it doesn't matter how hard you try, you can't bring it into focus anymore. So does this make sense, near point of accommodation? This leads us into the idea of presbyopia. Presbyopia, it officially means old vision. By the time you get into your late 40s and 50s, the lens becomes less flexible. Which means if I want to look at this tree, which is far away and the light rays are coming in parallel, it doesn't matter because the lens just is at its normal resting. So if I'm trying to look at this tree far away and I am 50 years old, it still looks like it always looked. What happens if I try to look at this flower, which is really close? And my lens, what does my lens have to do in order to make the flower come into focus?
Become more convex.
It has to become more convex. But now it is less flexible and it has less of an ability to accommodate. So the nearest that I can bring something and still have the ability to bring it into focus is a lot farther away than it used to be. So basically, your near point starts getting further and further away, right? Right, I'm sure you've seen this, like, a kid, low light in the restaurant is like, oh, I'll have this and this and this. And you've got somebody in their 50s that's like, what am I going to have on this menu here? Let me see. Like, the older you get, the farther something has to be away to keep it in focus. Right? I'm not, a lot of you are pretty young. You probably haven't started hitting it so much yet. Like I can say by the time I was in early 50s, it was incredibly frustrating to realize that things that normally you just look at, you don't see, that's why I have these. I never had glasses ever in my life. And then around early 50s, I have cheaters now because I can't see things that are a foot away clearly without something to help bend the light more for me. The rest of the vision's fine, but that near point of accommodation has gotten farther and farther away. In our lab, I would like each person to find their own near point of accommodation. All you do is get something. It has to be something that is clear to be able to see. Shut one eye and bring it farther and closer, and you'll find the place where you can't keep it in focus anymore. And just record, probably have a friend or a relative record the distance between your eye and the thing when it's no longer able to keep it in focus. We have data just to give you a sense. This is something I do want you to do it on yourself. I also normally we record everybody's data and put it in our lab book. Instead, I'm going to have you So do at home, record in your lab book, also discuss the group data. So here's near point of accommodation data from a previous semester. And if you take a look, what do you notice here?
The older the people get, the further away their near point is.
Yeah, exactly. So this is illustrating presbyopia very clearly. It's just, as you get older and older, the near point starts just getting farther and farther away. So when you do this, don't wear reading glasses, because reading glasses are actually designed to get around this. But if you wear like regular corrective lenses just to bring you to 20/20, you should be wearing those, right? Because if everything's blurry, it's not going to help. You're not going to be able to tell the near point. So in your lab write-up, determine your own near point, and also make sure you bring in this kind of group data and discuss what this means as well as we look at near point versus age and all that. So that's near point. And then we should talk about things like farsighted, nearsighted, presbyopia, hyperopia, right? So far we've just talked about the emmytropic eye, the eye that is functioning properly. It's like kind of normal. And we're going to see normal. There are eyes that are actually better than normal. It's not that uncommon to have like 2015, which is better than 2020. So normal, it just means, as one of my friends once was saying, normal is like, just because something isn't as common doesn't mean it's like weird. Like having red hair or blonde hair is not normal if you think about most people have brown hair, but it doesn't make you a freak. It's like, it's totally within kind of what you find out there in the population. But when we talk about normal, like what would be the preponderance like of what vision would be? So first eye I'm going to talk about the myopic eye. Myopic or nearsighted eye. So one of the ways to think about myopia or myopic eye is imagine it's like the eye we've been talking about, except it's kind of elongated. Trying to draw it kind of more elongated. Here's the lens. Here's like the retina. Let's say we're looking at the tree out here. The rays are coming in parallel. The lens bends it the way it would normally bend it, but because the eye is elongated, the retina isn't there. And then the rays cross and start diverging again. So what we see on the retina is blurry. So this tree is blurry because the lens is doing its normal bending that it normally would do, but that's like actually too much because this is far out here. Is there anything this person can do to bring the tree into focus?
Could they move closer?
All right, but let's say without moving. They want to see this thing far away. Yeah, the answer did. That
Do
would
they
work.
squint?
Right, and so and there is something about actually decreasing the aperture that increases the depth of field. So that actually could help as well. But let's just say, using the lens, is there anything that they could do with their lens?
They could accommodate it. Can't they make it more convex?
If they made it more convex, it would just bend it even more, which would make the problem worse.
Oh.
Right? So, and you don't, at this point, we're as flat as we can be. So basically, there's nothing you can do. Without glasses, this person is never going to see that tree in focus. But when we start looking at things that are closer, you know, if I look at my flower again, which is up close, and I said the flower has the rays that are already diverging to start with, therefore they are landing further back because they are getting bent the same amount, but that means they don't meet until about right here. Like this flower, actually, you can see if something's close and the rays are already diverging to start with, then I can still bring it into focus. But I'm never going to be able to focus something that is far away because I don't have the ability to diverge the lens, the light even more. I can only converge the light more with my lens. So this is nearsighted. If I'm nearsighted, I can see things that are up close because of this optics, but I can never focus on something far away, even if I try. The hyperopic or farsighted eye is kind of the opposite. Here, like the eye would be like too short. So I'm looking at my tree and the light rays are coming in parallel. Again, normally they would converge here, but the place where the retina actually is, they're not in focus, so this tree is blurry. Is there anything this person can do to bring the tree into focus? Well, what does the lens do if it wants to? What can we do with the lens? I can't hear anybody.
Well, can the lens become larger so that it converges tighter?
Yeah, so probably larger is not quite larger, but it can be more curved, which means we're gonna pull the light in, bend it more, and we can bring it into focus. So it's possible to bring this tree into focus, but it means that we actually have to use the lens. Normally, if you're looking at something far away, the eye is just relaxed and something far away will be in focus. If you are farsighted, you have to accommodate to bring something far away into focus. So I actually have to work. My eyes are getting tired just trying to keep this tree into focus far away. Normally, your eyes would not have to be working to keep something far away in focus. But it means now we've kind of blown our wad with this accommodation. We're using the accommodation just to make the far thing look regular. When we start to try to focus in on something closer in, where normally you would accommodate because the light rays are coming out, we've already done all we can do. There's not more curving left in reserve, so we can't ever bring the near stuff into focus. Right? That's why this person is farsighted. They can see things that are far away, but they can't, because they've already used their accommodation just to bring the far thing into focus. There's no reserve left to do the normal accommodation, which would be bringing even the closer stuff into focus. Does that make sense? In both of these cases, you can fix this with a corrective lens that just makes it like the emmetropic eye. If you are a myopic person, what you need is a lens that will diverge the light before it hits the eye, so when your eye is relaxed, it lands on the retina. What kind of lens are we going to use if we want to just diverge the light a bit so it's going to land further back?
Sorry, can you say that one more time?
So I have the light coming in, and right now when the light comes in, it doesn't bend. I mean, it bends too much, so it's converging before. I want to put some kind of lens in the way here that's going to diverge the light a bit, so it's going to land a little further back on the retina and be in focus.
So you need a concave lens.
Exactly. So corrective lenses for people with nearsightedness are concave. The light comes in, it goes out a little bit right before it hits your eye, and then when it goes through the lens, it lands exactly where it's supposed to be. So this person is now the same as an emmetropic eye, and everything's working like it would be normal. You know, they'd be 20/20. And we're gonna, normal. When you do the acuity testing, whatever your vision is without glasses, when you put your glasses on, it should usually end up being 20/20 because they're designed to bend the light just right so it lands on your retina when you're just looking at something far away, and everything works like normal. You know, for the far-sighted eye, we have the opposite problem. The light coming in is converging too far away at normal, so we're going to put a convex lens in there. And now the light coming in is going to bend in a bit, and by the time it's done with the lens, it's going to land right where it's supposed to. So far-sighted, you're going to have to pre-bend the light in. Nearsighted, you have to pre-bend the light and diverge it a bit, so it'll actually land on the retina without any accommodation. Right, and it's really easy to tell. If you have some friend and you're wondering if they have farsightedness or nearsightedness, if you just take their glasses and hold it over a page, this is a magnifying glass. If their glasses act like a magnifying glass, then you know they're farsighted. This makes everything look smaller. If you hold their glasses in front of a page and all the letters look dinky, you know that they're nearsighted because they need a concave lens in front of their eye to make everything normal. So, are there questions about this? So when we want to test somebody's visual acuity, the way we do it, what I'm going to do is I'm going to kind of show you the way it's normally done, but because we're at home, we're going to do a slightly different version of it. So you only use one eye at a time. You stand 20 feet away from this chart. It's called the Snellen eye chart. You know, and it has different rows. E, like, I don't know, A, B, blah, blah, blah. With smaller and smaller letters. Everybody has seen this. So you're standing 20 feet away, and you read out each row. You're saying, okay, I read out that row, it's an E. The next row. And you keep going until you find what is the last row that you can read. You get totally correct. If you miss even one letter on that row, then it doesn't count. You have to find what is the last row that you get totally correct. And as you go down the list, each of these will be like, 2200, 2100, 2030, 2020, 2015. It tells you these two numbers. That is your vision, visual acuity. And the two numbers are this is basically you now. This is Emmytropic Eye. So if you are 20/20, that means you standing at 20 feet see with the same clarity as someone with an emmetropic eye standing at 20 feet away from the chart. So 20/20 means your normal vision. If someone was 20/40, what would that mean?
They're the same as somebody standing 40 feet away with an emmytropic eye?
Yeah, exactly. So you're standing 20 feet away from the chart. You see with the same clarity of someone with normal vision that's standing 40 feet, twice as far away, they see what you're seeing when you're 20 feet away. So your vision's worse. You have to stand twice as close to see what someone with normal vision sees. It's not uncommon for people to have like 2100. When they take their glasses off, everything is just totally blurry. You're standing 20 feet away and you see as clearly as someone standing 100 feet away. It's not uncommon in our class, particularly with kind of people with young, healthy eyes, to end up with like 20/15. What would 20/15 mean?
Your vision is the same as someone with an emmetropic eye standing 15 feet away, so you have better vision than
Exactly. You're standing 20 feet away and you see just as well as normal person vision standing like 5 feet closer to you. So you can be 5 feet further away and still see as good as somebody with 20/20 vision standing, standing 15 feet away. So make sure when you are testing your eye and writing down, what you are, 20/20 or 20/40 or 2100 or whatever. And there's no shame. I know a lot of amazing people who are like 2200 and stuff and just wear glasses and bring them to 20/20. So you're going to test, and you do it one eye at a time. Do your right eye and find out what that eye is, and do your left eye and find out what that is. If you wear glasses, you want to do it with and without glasses. See what your vision is like without them, and then almost always once you put your glasses on, if your prescription is up to date, it should bring you up to 20/20. For our class, instead of using a normal Snellen eye chart in 20 feet away, which is kind of a pain in the butt, I think Tina found this thing. Visual home acuity. Snellen eye chart for 6 feet away. Test your vision at home. So basically you can use a PDF or the other document You know, and you can basically take this thing and you can print it out, you know, and read it and have somebody like double checking it for you as you read it. And again, it says here, print it out, make sure that the first large E is about 2.5 centimeters tall. Then tape the chart 6 feet away and find out what it is, what you have for your right eye, for your left eye, without glasses, with glasses, standing 6 feet away, looking at this little printed out chart. Again, here it kind of, this one actually only goes to 2020. If you can read this last line here without any mistakes, it's 2020. Don't memorize it before you put it up there. That's just not making, you know, being that useful. You know, normally if we're in class, we use a more complex chart, and you have to stand 20 feet away, but that's kind of tricky to do inside your living room. So that's why we have this thing for you for, you know, so after class or after we're done talking, print this out. This is something you're going to have somebody help you with because you can't actually know if you're making a mistake. So that is the main way we test visual acuity, though. And also, this is a Snellen eye chart. Like if there's some question, how do we test for visual acuity and what does 20/20 mean? You should know both of those. You should know what 20, if I'm 20/70, you should know what that means. The other thing that we look for in visual acuity is what's called astigmatism. Like you've probably heard of that. Like your glasses usually correct for kind of your visual acuity, like make sure you're 20/20, but then there's this other thing called astigmatism. Astigmatism, like everything we've been talking about so far, is assuming that the eyeball is perfectly spherically symmetric. Like this distance and this distance and this distance and this, everything is exactly symmetrical in this kind of radial symmetry. Sometimes that's not true. Sometimes the lens or the cornea is more like of a football, in which case light coming on one axis might actually bend differently than light coming in on another axis. So it could be I'm looking at something and the light from this way lands on my retina, but the light from this way is landing in front of or behind my retina. So astigmatism is when there is some non-spherical symmetry in either your retina or your cornea, not your retina, your lens or your cornea. And so light coming in from one orientation actually gets bent slightly different from another orientation. And to test for that, we use this thing that's an astigmatism chart. It looks kind of like a star. It's right here, this thing. The basic idea here is, again, this is another thing you can just print out and put on the wall and look at one eye at a time. And if your eyes have no astigmatism at all, every axis, 12, 6, 3, 9, 1, 7, all the lines should look exactly the same. The exact same thickness, the exact same clarity. If you have astigmatism, then one axis will look different from another. Some will look darker or bolder or blurrier. Like I know when I look, like 12 and 6 are really clear, but 1 and 7 look kind of thinner. And it doesn't matter. If I turn my head this way, 3 and 9 look clear now, right? It's not about the actual axis. It's about the axis with respect to your eyeball. So do this as well at home. You're going to look at, is there astigmatism? Look on your right eye, your left eye, describe your observations. And then also, again, describe what it means. What is astigmatism? What would it mean if you have or don't have it? So those are kind of the visual tests that we're going to be doing. The last thing we're gonna talk about for this lab is binocular vision. The fact, that you have two eyes. Right? Having two eyes, it is kind of nice. If you accidentally poke out one eye, you still got an eye. But what do we really gain from having two eyes over one eye? What's one of the really important benefits of having binocular vision?
Is it our field of depth?
It's being able to have depth perception. So not so much the field of depth, like the field has to do more with kind of how big the iris and everything, like how much is in focus all at once. But whether or not you can detect, like have this visceral sense of far away and close up, really depends on having two eyes. Like if you just shut one eye and look around, everything looks weirder. It looks kind of flatter. If I put one hand over one eye and just kind of look around and just notice that everything, I don't see people doing it. You should do it. Put one hand over your eye, and look around. Try to get a sense of depth. What's closer, what's farther. Everything just feels flatter. Then you open your eye and all of a sudden it's like, oh, that feels so nice. Everything kind of breathes again. I don't know if you noticed that. Right? Because each eye has a slightly different view of the world. If you you've probably done this, you put a finger in front of your eyes and you blink back and forth and back and forth and back and forth, your finger jumps all over the place because each eye is actually having a slightly different view of the world. Everything's kind of oriented slightly different from each other. And your brain makes these calculations based on that information to create this experience of stereoscopic three-dimensional vision. Right? Because if you look at a person, both eyes basically are looking at the same thing, but they see slightly different versions of it, and you can use that information to get this three-dimensional information. Animals that tend to be more predatory, like a cat, has its eyes in front of its face because it's important, like, where is that mouse? I need to pounce. Or if you're maybe an owl, you want to also, you're getting that mouse. You're ready, boom, you got dinner, right? So you need to know exactly how close is this thing so I can attack. On the other hand, most animals that tend to be more on the other side of this equation, like the mouse, they tend to have their eyes on the side of their head, right? If you're a mouse and your worry more is about becoming dinner rather than catching dinner, your eyes are on the side of your head and the overlap is very little. So the good thing, you don't have good depth perception. In fact, mice have pretty crappy vision, but it's hard to sneak up behind a mouse or a bunny rabbit for that matter. If you have a bunny rabbit, a cottontail, or like, one of the little, not a predatory bird like a hawk, but more of a little songbird. Most of these little guys that tend to be more hunted than hunters tend to have their eyes more to the side. So then they have kind of 360 degrees of vision, so you can't sneak up on them, but they don't actually have that good three-dimensional. But that's not as important to them. They're more concerned that you don't get sneak up on and eat them. So in our lab, I say to use a test tube, but you can use any little thing. This is another thing I would like you to do at home. Where basically you have one person hold something, and it should be, and then here's you like with a pencil. And you basically want to sink it in there. And if you use two eyes, versus just one eye and see how well you do it. And again, if you don't have a test tube and a pencil, this you'll come up with something. You could even just have, you know, take some little paper and make some little target, right? And then have a, some pencil and, and make sure it's far enough away that I could. Make sure it's far enough away that it's not right there and they're on top of it. Make sure it's not exactly the distance of their outstretched arms, so they have that. So, and then make sure that they're not like going, you want to just be kind of bold, like sink it in, rabbit in the hole, and do it with both eyes open. And then shut one eye and like rabbit in the hole. You're probably going to see there's a big difference. So do it and record your data in your lab book. So that's kind of the last piece of the lab, is just kind of really getting a sense of how having two eyes in one eye really can make a big difference in depth perception. Is there anything else that helps you with determining depth besides having two eyes and the stereoscopic vision?
Would it be seeing color?
Not so much color, although I guess you could say like things farther in the distance tend to be more kind of faded. Actually, that's true. Like if you've been watching like Bob Ross or something, you're trying to draw trees in the background versus trees up close, you'd kind of make them be more a little gray or something. So yeah, no, that would be a clue. Things farther away tend to be a little more hazy and faded. Yeah. What else? What other things?
Is it our ability to change the shape of the lens, like our ciliary muscles to?
So that's going to be more about focusing rather than determining depth of something. Yeah, so I would say that wouldn't really be as much of it. So clues, information, cues that you have. I mean, there can be blocking. If something is hidden behind something else, that thing that is blocking is probably closer than the thing that's blocked. And then another big, and how things move with respect to each other. As you move around, things are going to like change their relationship to each other, and things that are closer are going to move far more dramatically than things that are far away. And then a huge one is perspective, right? If you look at somebody right in front of you, their head is pretty big, filling up your whole field of view. You look at somebody across the room, their head's kind of small, but you're just assuming like, okay, that person didn't have their head shrink. They're just far away. That's why their heads are small. Whereas, so this idea of perspective is huge. And like I gave an example in the, here, here it says, in your discussion, also talk about what the picture below shows us about how we determine depth. So does everybody see this picture on their screen right now? I mean, what does it look like?
That car is giant.
Looks like a giant car. I want you to like use your fingers or whatever else you have to actually check the size of each car. Like just measure from tail light to tail light or whatever.
It's about the same.
It's not even, it's exactly, it's just a Photoshop. They're just cut and pastes of each other. So this is showing like this idea of perspective. You have this kind of built-in kind of program in your brain that if something is farther away, it's going to be smaller. So if you have a car, like the same size car in reality, but because it's, the perspective of the whole street makes it look like it's farther away. If something looks that big and it's that much farther away, it must be huge to still be that big. Whereas the one closer, right? So this is just an example of how your brain is actually pulling in all this information to determine what's closer or farther. It's not about using two eyes. This is about perspective, how lines that get farther away tend to kind of converge, right? That was a huge advance in just art. Like if I'm trying to, here's a road going off into the distance, it's like, I'm not actually thinking that, oh, the road is getting skinnier. It's like the road's just getting further away. And as the further it away, the things just kind of converge and go off into a point, right? That's perspective. Your brain is just knows that and takes that and uses that information. So if I see everything else, the road is converging, there's all these clues that this is just far in the distance. And then I have something here that's a certain size and something here that's the exact same size. In order for this to make sense, this has to be huge compared to this. If all the ratios stay intact, right? So just giving you a sense, there's other cues we use other than stereoscopic vision to determine distance. And some people are better than others at using stereoscopic vision. Some people don't do it very well at all. Some people do it well. So, but anyway, blah, blah, blah. Pupil. What is the pupil? As you look into the eye, what is that black dot?
Isn't it just a hole?
Yeah, that's just a hole, right? Just kind of making sure people, right? Here's the light coming into the eye, through the lens to the retina. You have the iris, which makes a little hole for the, more or less light to come in. But it's just dark in here. There's no light in here. It's dark. So you're looking into a cave, it's going to look black. But if you take a flash photo of somebody and all of a sudden there's light coming in and illuminating the back of the eyeball, you see all the blood and everything. And it's like, dag nabbit, I got red eye, right? You're just actually just seeing the inside of the eye because now you're illuminating it with the flash. But the pupil is actually just like the hole looking into a dark cave. Okie dokie. Any comments or questions about the lab? We are not doing the ophthalmoscopic exam, so no part 8. But I think you'll probably have fun with these, especially if you have somebody who's willing to kind of be game at home, like doing the little eye exams. And actually, especially if you wear glasses, it can be cool to actually see what is my vision without glasses and with glasses. And really get a sense of, oh yeah, they are designed exactly to return me to 2020 and all that.